Welcome back everyone. I am Saptaparni from BioLive. Hope all of you are doing very well. So in today's video we are going to learn about types of lipids or classification of lipids. Okay. This video is going to be very useful for any students who are undergoing UG or PG courses in biochemistry or zoology or botany, and also for those students who are preparing for NET examination or NEET examination. Okay, so see here, there are three types of lipids. The first one is simple lipid, which is made up of only fatty acid and alcohol. Okay, so simple lipids are the esters of fatty acid and alcohol. Next, compound lipid, which is a ester of fatty acid plus alcohol along with other groups. Okay, these groups can be phosphate group, nitrogenous or non-nitrogenous groups, amino acids, okay, or glycerol even. Then derived lipids. Derived lipids are hydrolyzed product of lipids like fatty acid, alcohols like glycerol or sphingosine, or sterols and steroids. Okay. So these are three types of lipid, and now one by one we will go into depth of each type. So first of all, let's dig deep into the simple lipids. Simple lipids are simple esters of fatty acid and alcohol. Now this alcohol can be glycerol or any kind of higher alcohol. Okay. When the alcohol is glycerol, then those kinds of simple lipids are known as glycerides. Okay. Now fatty acid can be, as we know, there are three hydroxyl molecule in glycerol, and these three hydroxyl molecule of glycerol can be esterified with either 1, 2 or 3 fatty acids. When there is only one fatty acid present, then that type of glycerides are known as monoglyceride. When two fatty acids are present, that is known as diglycerides, and when three fatty acids are present, that is known as triglycerides or fats. So okay, so this is the difference between lipid and fat. Fat is a type of lipid, but lipid is not a type of fat. Okay, the triglycerides are generally known as fat. Now these triglyceride can be of two types. One is simple triglyceride, another one is compound triglyceride. In simple triglyceride, three fatty acids which are esterified with glycerol's hydroxyl group are same in nature. Okay, whereas in case of compound triglycerides, the fatty acids are different from each other. Okay, it can be two fatty acids are of same type, one is other type, or three fatty acids all are of different type. Okay, now see an example here. See, this is the glycerol. This is the formula of glycerol, and this carbon is known as alpha prime carbon, this is beta prime carbon, and this carbon is alpha carbon, and these are the three hydroxyl groups of glycerol. Now see, when three fatty acids are same, all the fatty acids, three fatty acids are same, R1COH, R1COH and R1COH. These three fatty acids are going to react with three OH groups of glycerol. Okay, and from here the OH group of COOH and the hydrogen of OH group will be liberated as H2O, Okay, so similarly from these layers also H2O will be liberated. So that is why 3H2O will be liberated, and the glycerides that will be formed will be of CH2OCOOR1, and this is the ester linkage. Okay, this is the ester linkage, and as all of the three fatty acids are of similar type, this triglyceride is known as simple triglyceride. Okay, now coming to the mixed triglycerides, see here all the three fatty acids are different. This is R1COH, this is R2COH, this is R3COH. So all the fatty acids are different, that is why it is a mixed triglyceride. Now these simple and mixed triglycerides actually form the oil or liquid type of fat that we can see at room temperature. Okay, now there is another type of simple lipid that is known as wax. In wax, you can see there are fatty acid, obviously there are fatty acid, but those fatty acids are quite long in chain. Okay, C16 to C34 carbon containing fatty acids are there, which forms the ester bond with long chain aliphatic or alicyclic monohydric alcohol. That means in these type of alcohol, unlike glycerol, there will be only one hydroxyl group, one OH group. In glycerol, we know there are 3 OH group. Okay, so see the wax. Here the long chain fatty acid like palmitic acid. Palmitic acid is a C16 fatty acid. Okay, the formula is C15H31COH, and here the long chain alcohol is C30H61OH, which has only one hydroxyl group, as I have told earlier, and this is the miracyl alcohol. When this palmitic acid and miracyl alcohol reacts with each other, obviously from here the OH group, and from here one hydrogen group will be liberated as H2O, and the bond will be formed as C15H31COO C30H61. Okay, sorry, from here hydrogen, and from here OH group will be liberated. OH group will be liberated, and the thing will be formed is miracyl palmitate, which is beeswax, which is a component of beeswax. Okay, so beeswax or wax is the ester of long chain fatty acid with long chain monohydric alcohol. Okay, got it? Now these are the types of simple lipid. Now coming to the main part, the compound lipids. What are compound lipids? Compound lipids are those which are esters of fatty acid and alcohol along with other groups. Okay, so when the other group is phosphate, then that type of compound lipid is known as phospholipid. When the other group is carbohydrate, then that type of compound lipid is known as glycolipids, and when the other group is protein, then that type of lipid is known as our lipoproteins. Okay, so there are 3 types of compound lipids: phospholipid, glycolipid, and lipoproteins. We will come to each one by one. Okay, so see, in case of phospholipid, you can see these are the esters of fatty acid and alcohol. This alcohol can be glycerol or sphingosine. Okay, and then with this fatty acid, with this alcohol, there will be a phosphate group attached. That is why the name is phospholipid. Okay, and along with this thing, when fatty acid, alcohol plus phosphate group is there, there could be some nitrogenous or non-nitrogenous substances also. Okay, so let's take a look one by one. So phospholipid can be of 2 types. One is phosphoglyceride and second one is sphingomyelin. So in case of phosphoglyceride, you can see the alcohol is glycerol. Okay, the alcohol here, the alcohol is glycerol, and these 3 carbons of glycerol are esterified with, these 3 positions are esterified with, the alpha prime carbon is esterified with saturated fatty acid. Saturated fatty acid are those fatty acid which have no double bond in them. Okay, then the beta carbon is esterified with unsaturated fatty acid with double bonds. Okay, 1 or more double bonds, and the alpha carbon is saturated with esterified with forms the bond with phosphate group. Okay, phosphate group, and this thing, the glycerol plus all these things at each position forms the phosphatidic acid. Okay, and when with this phosphatidic acid some nitrogenous or non-nitrogenous groups are attached, then we can call it is a phosphoglyceride. Okay, why it is called phosphoglyceride? Because here the alcohol is glycerol. Now let's take a look on what kind of nitrogenous substance could be attached with phosphatidic acid. When phosphatidic acid binds with choline, then we can find phosphatidylcholine or lecithin. Okay, choline is a nitrogenous substance. When phosphatidic acid reacts with ethanolamine, which is also a nitrogenous substance, it forms phosphatidyl ethanolamine, or another name is cardiolipin. when phosphatidic acid reacts with hydroxyl amino acids like serine, threonine, hydroxyproline. Hydroxyl amino acids are those amino acids which have additional OH group, okay. Then they form phosphatidyl serine or phosphatidyl threonine or phosphatidyl hydroxyproline. If you have read about the structure of plasma membrane, you can find all these phospholipids in our plasma membrane. Phosphatidyl choline, phosphatidyl ethanolamine, phosphatidyl serine. Okay, this name will be very familiar to you. Okay. Next, plasmalogens. What are plasmalogens? Plasmalogens are one kind of phospholipids. We can call it as ether phospholipid. Why it is called ether phospholipid? Because see, at the beta carbon position, obviously like the previous one, unsaturated fatty acid is esterified. At the alpha carbon position, phosphate group and choline, okay, choline, ethanolamine or whatever there will be in that. But at the alpha prime position, that means at the first carbon of the glycerol, there will be alkene instead of saturated fatty acid, okay, and this alkene will form a ether linkage with the glycerol, okay. So that is why this type of phospholipids are known as ether phospholipids or phosphatidylcholine. Let's see here. Previously, whatever we have read till now, we have seen phosphatidyl serine, phosphatidyl ethanolamine, phosphatidylcholine, but here it is phosphatidylcholine, okay. Now coming to another type of phospholipid, which is non-nitrogenous substance containing phosphoglyceride. Here see, phosphatidic acid is esterified with non-nitrogenous substance like a vitamin inositol and it forms phosphatidyl inositol. Then it can also get esterified with another molecule of glycerol, then it will form phosphatidyl glyceron, or 2-phosphatidic acid can form ester bonds with another extra molecule of glycerol, and that will be called diphosphatidylglycerol, okay. So these are the different types of phosphoglycerides under phospholipid, okay. Now in case of phosphoglyceride, we have seen the alcohol is glycerol, but when the alcohol is sphingosine, that type of phospholipids are known as sphingomyelins. What is that called? Sphingomyelins. So sphingomyelins are also phospholipid, but here the alcohol is sphingosine, not glycerol, and sphingosine is a C18 nitrogen containing and 2-hydroxyl group containing alcohol, okay. So see here, there are 18 carbons are here in sphingosine. There is a NH2 group, okay, and 2 OH groups. See here, this is a OH group, this is a OH group. When this sphingosine, this alcohol, C18 alcohol, reacts with long chain fatty acid, this is the fatty acid, okay, here this is the fatty acid, CH3 and whatever the backbone, and here COOH, they form a CONH bond, a amide bond reacting with this NH2 group, okay. The long chain fatty acid reacts with this NH2 group and water is liberated and a amide bond is formed here, which is CONH bond is formed here, and this thing, when sphingosine is bonded with your long chain fatty acid, that is known as ceramide, okay, that is known as what? Ceramide. And now this ceramide can form ester bonding, bonding with phosphocholine or whatever, okay. When phosphocholine is linked here at this CH2OH position, okay, here this CH2OH position, then the entire molecule is known as sphingomyelin. What is sphingomyelin? Okay, sphingomyelin is also present in our plasma membrane, okay. So these are the two types of phospholipid. One is phosphoglycerides and another one is sphingomyelin. In phosphoglyceride, the alcohol is glycerol, and in sphingomyelin, the alcohol is sphingosine, okay. And phosphoglycerides can be of various types depending on their presence of nitrogenous or non-nitrogenous groups. Now the second type of compound lipid, which is very important one, is glycolipid. Here the ceramide. What is ceramide? When sphingosine, the alcohol sphingosine forms an amide bond with the long chain fatty acid, that is known as ceramide. So this is the ceramide. Okay. When ceramide forms bond with some kind of carbohydrate, that can be monosaccharide or oligosaccharide, that type of lipid is known as glycolipid. And here the carbohydrate forms bond with the CH2OH of the sphingosine. Okay. And here the bond is a glycosidic bond. Okay. Now, glycolipids can be of different types. The first one is cerebroside. Here you can see the carbohydrate is monosaccharide. It is either glucose or galactose. When the carbohydrate is glucose, it is known as glucocerebroside. When the carbohydrate is galactose, it is known as galactocerebroside. Okay. So ceramide plus glucose or galactose forms our cerebroside. Now, what is sulfatide? Sulfatide is ceramide plus sulfated glucose or galactose. Okay, same like cerebroside, but here the glucose or galactose is sulfated. Now coming to ganglioside and globoside. Now the third galactolipid is ganglioside. Here you can see the ceramide is forming bond with branched oligosaccharide which contains nana or sialic acid or N-acetyl neuraminic acid. Okay, so obviously here ceramide will form a bond with branched oligoside. See here, this is the ceramide and it is forming a bond with the branched oligoside. And in this branched oligoside, here you can see the branching, here is the branching, and here it will obviously contain a N-acetyl neuraminic acid or nana or sialic acid. Okay, so ganglioside always contain nana or sialic acid, and other moieties can be glucose, galactose, N-acetyl, galactosamine, galactose, etc. Okay, now coming to globoside. In case of globoside, you can see ceramide is there and the carbohydrate is oligosaccharide without nana. Okay, so here there will be also oligosaccharide but no nana will be there. Okay, so you see here, this is the ceramide and with it there is a oligosaccharide containing glucose, galactose, N-acetyl, galactosamine but no nana. Okay, so these are the four types of glycolipids which forms the glycosidic bond with ceramide. Okay, now coming to the third type of compound lipid, which is lipoprotein. From the name it is very evident that here the lipid will be conjugated with protein. Okay, lipid and protein both will be present there. So see here, this is a general structure of lipoprotein where you can see a globular ball-like structure. Okay, this is a globular ball-like structure which contains a monolayer of phospholipids. Okay, phospholipids or triglycerides. These are the polar head group of those triglycerides and these are the non-polar tail groups, okay, of those triglycerides. And along with these these monolayer of amphipathic triglyceride, phospholipid or cholesterol esters, you will also find some proteins. Some will be integral proteins, okay, and some will be peripheral proteins. Integral apolipoprotein is of ApoB100 and peripheral apolipoprotein will be of ApoA, ApoC, ApoD, ApoE, etc. Okay, and in the core you will find non-polar lipid rich central core region. Okay. Based on its density, okay, based on its increasing density, we can divide it into various groups like chylomicron, which has the lowest density, then VLDL or very low density lipoprotein, LDL low density lipoprotein, IDL intermediate density lipoprotein, HDL high density lipoprotein, and albumin FFA. These are different types of lipoproteins, and in our coming video we will be learning the function of each of them. Okay, so here you can see from the upside down the density is increasing, high density lipoprotein. Okay, and when the density is increasing, that means the lipid content and its diameter is decreasing along with its flotation rate. Okay, just remember that when the density is increasing, that means the lipid content, diameter and the flotation rate of these lipoproteins are decreasing. Okay, increasing density is correspond to decrease in its lipid content, diameter and flotation rate. Just remember that. Okay. Now coming to the derived lipids, the third type of lipids, that is derived lipids. Derived lipids are all those lipids which are formed from the hydrolysis of lipids, like any types of fatty acid, that could be saturated, unsaturated, glycerols, sphingosine, that means any kind of alcohol, and sterols and steroids. So here we will be focusing on sterols and steroids. What are these? Steroids, first of all. So see here, the steroids are composed of 4 rings. 3 of them, see here, A, B and C. These 3 rings are cyclohexane ring, and this last ring, the D ring, is a cyclopentane ring. So it contains cyclopentano parhydrophenanthrene ring or nucleus. Okay. And here there are total 17 carbons in this ring structure. So any steroid, you name it, it could be progesterone, estrogen, testosterone, there will be this nuclear structure with various substitutions. Okay. So steroids can be of 2 types. One is normal steroid, another one is allosteroid. When the A ring and B ring are in cis condition, that means they are in the same plane, and the 5 prime and the hydrogen at the 5th carbon and the methyl group at the 10th carbon is also on the same plane of these 2 ring. Okay, then that type of steroids are known as normal steroid. Okay. And when these 2 are in the same plane, we draw a straight line here. Okay. But when these 2 rings, A and B rings, are in different plane or in transposition, okay, and these C10 methyl group and C5 hydrogen group are in different plane. See, this one is below the plane, and this one is this one is above the plane, this one is below the plane. Okay, then that kind of steroids are known as allosteroid. Okay. Now from another point of view, we can also define steroid or classify steroid into 2 groups. One is alpha oriented group, another one is beta oriented group. In case of alpha oriented group, you can find that the substitutions here, the in the 3rd carbon there is a substitution OH group. Here it is also same, but in case of alpha oriented substitution, this substitution group will remain below the plane of this ring. Okay, and in case of beta oriented, this substitution will remain above the plane of these 2 rings. Okay, so that is why name is alpha and beta. Okay, got it? Now the last type, sterols. Sterols are actually steroids with some modifications. What are those modifications? In case of sterols, there will be obviously a OH group at the 3rd carbon. See here, there is a OH group at the 3rd carbon, and along with that a side chain will be there. A side chain containing 8 to 10 carbons will be there on the 17th carbon. See here, this is the 17th carbon. Okay, this is the 17th carbon, and here you can see there is a side chain. There is a side chain. Okay, there is a side chain of C8 carbons. Okay. So this is one of the sterol, very famous sterol known as cholesterol. Here you can see it is named as 3-beta hydroxy because here the hydroxyl group is present at the 3rd position. Delta 5 cholesterol. Why delta 5? Because here you can see a double bond at the between the 5th and 6th carbon. That is why delta 5. Okay, cholesterol. And here there is a C8 side chain at the 17th position, and at this position C10 and C13 position there is methyl groups, methyl substitutions. Okay, so this is the overall structure of cholesterol, one of the famous sterols. Other sterols are like like lignosterol. Okay, these are other sterols. So this is all about the classification of lipids in a very short manner. You can refer to different books of biochemistry for more insight. Okay, I hope this video is a very gist video and this will be useful for you. And if you find this video useful, don't forget to hit the like button and subscribe to my channel for more such videos. Thanks for watching and happy learning.